Hi, Alex Gronchi, surgical oncologist from Milan. Have you noticed any differences between the different leiomyosarcoma subtypes? I mean, uterine, non-uterine, but also in the non-uterine, whether abdominal, extremity.
Mm-hmm. Yeah, so in our pilot study, there were both uterine and soft tissue leiomyosarcomas included, but it was a small study, so we haven't really identified any differences between them. Also, when we constructed our panel, we haven't seen any genomic changes that would be maybe more frequent in uterine or soft tissue leiomyosarcoma. So in that particular study, we haven't seen any differences.
I have a question for Lily. The osteosarcoma cell lines that are highly dependent on SMARCA1, what happens when you knock it down? I'm particularly interested in if you get stable lines and the knockdown is perpetuated rather than they find a workaround and re-express the SMARCA1, then have you been able to identify accommodations, compensatory mechanisms that would also give you a better sense of what pathways can either stand in or could be alternately targeted in a situation where you're able to do something about SMARCA1?
Yeah, it's a great question, Jonathan. I mean, we haven't done any very long-term knockout experiments, like, for example, knockouts in mice, which would help us to be able to see what the breakthrough mechanisms might be in those tumors that eventually grow out. Those studies are ongoing. But we do see that the cells, they do mostly just die. They're not present on, they're not senescing or hanging out on the dish. They're really dying. I do think that signs point to potentially some of these DNA damage inhibitors or DNA checkpoint inhibitors such as ATR, CHEK1, potentially WEE1 being really helpful, potentially synergistically with this, although there's no drug targeting SMARCA1 yet, but that's something we're potentially interested in.
Hello, I had a question about the circulating tumor DNAs. I was curious how you feel like your studies, I was particularly interested about the two different or more means of checking it, how that compares with some of the clinically available tests now, like Signatera.
Yes, so as everyone is probably well aware, there are already commercially available circulating tumor DNA tests that are in use. I think what is particular for our approach is that we want to develop a disease-specific test. The tests that are already available, they use also panels. They target selected mutations, but these are usually mutations that are very frequent in different types of cancer. They were developed mostly for several most frequent types of cancer. And while leiomyosarcoma shares some of those alterations like P53 or mutations in some tumor suppressor genes may be common for different types of cancer and leiomyosarcoma, there are many different types of alterations that may not be covered by those tests. So this is how I see that focusing on the disease and developing a disease-specific test may improve the performance.
For what it's worth, Signatera does 16 tumor-specific genes, so that's the first one I know of that's more tumor-specific, but I don't think it does that copy number alteration.
Okay, you mean the patient-specific approach?
They sequence the tumor, pick 16 genes, and try to track those in the blood.
Oh, yes. So actually, I think this is a great approach, and I'm also aware that my collaborators are applying this kind of approach. What I try to overcome in our study is that the way I see it is that if there is a panel constructed for a patient based on the analysis of their tumor at some point in time, then genetic evolution may occur, right? So there may be some new variants that may occur in response to treatment, for example, or with new metastatic lesions, and they may not be trackable anymore because the panel was constructed for a tumor that was, let's say, removed a few years ago. So this is why we also want to cover like a broader spectrum of alterations that can be found in the disease, both in primary and metastatic tumors, and this is how I see it's different.